Hello everyone. So today it's week 8. Last class I was already teaching like how the diversity in the antibodies, there is how much so much diversity is there in antibodies, and how each B cell expresses a specific B type receptor or a specific type of antibody which is specific to a particular pathogen, and how many and how many types of recombination can occur between the specific uh, like chains, the VDJ, and how heavy and light chains recombine together to produce a large number of molecules. So here I haven't finished the last class, so I will continue with the B cell as well as some like some of the left a portion of the B cell rearrangement, then I will go to the T cell rearrangement. So here you can see, like I have already told, like B like antibodies are composed of heavy one, like heavy chain as well as light chain, and it is having variable region as well as constant region. So there are two types of light chain, a kappa as well as lambda. And in case of the heavy chain, there are five different types which can give rise to the five different molecules of antibodies. So, however, it is like uh, we know that each chromosome is having like two alleles. So, if there are two alleles, so it will produce a similar type of antibody if in from both the allele it is producing. So to uh, like bypass that, each B cell synthesizes only one heavy chain and one light chain. Otherwise, there will be four kinds of, like one heavy chain will recombine with another light chain, and then it will combine of many types of antibodies. But each B cell should represent only one type of receptor on its surface. So from one allele, only one light chain and heavy chain will be produced. So B cell express immunoglobulin heavy and light chain genes from only one allele, which is a term known as allelic exclusion and one light chain gene family. And heavy chain rearrangement occurs first after before light chain rearrangement. And in mice, kappa chain rearrangement occurs before that of lambda chains. And the generation of this functional B cell receptor is very much energetically expensive because of the considerable wastages involved to reduce the number of non-productive gene rearrangements. Right here you can see a diagram. For example, this is a progenitor B cell. So it needs to produce its receptor on the surface of this cell. So what happens first, as you all know, in case of heavy chain, there will be VD and J and C regions. Four regions will be there. And in case of light chain, there will be V, J and C regions. And this first, in case of heavy chain, as I already told, heavy chain will occur, rearrangement will occur first, followed by light chain rearrangement. So here, first what happens? There is a recombination between DH and JH. First, because V chain, the rearrange will be first V segment will come, then D segment, then J segment, then C segment. So there will be a recombination between D and J segment. So here you can see uh, first it is having the DJ rearrangement. After that only V will combine with D and J. So either it can form a productive allele, like here VDJ can recombine to form productive allele, and here you can see the receptor, or otherwise it can form a non-productive allele. And here you can see like this VDJ, when it is joining, it is forming a non-productive allele. The cell will again try to recombine, correct itself to minimize the wastage second time. Here you can see again it is trying to recombine to form a productive allele, but again it is failing, it is forming a non-productive allele, and then what happens, this leads to cell death. So the cell dies because it is unable to produce a productive allele. So here you can see it is forming a productive allele, and if it here also if it forms a productive allele, then this will further go into the light chain rearrangement. So here again in case of the light chain rearrangement, V and J will join in both the cases, and what happens here you can see again it can form a productive allele where all the joining has taken place correctly, and it is for having a IgM. But in case of this second one, again it is forming a non-productive allele. So it cannot form that receptor on its surface. It again tries to form a productive allele. it can either try and form another productive allele, or it can again lead to a non-productive allele. So here, this mu plus k means the IgM is formed here also, and but here it is a non-productive allele. So again, it is trying once again to form a productive allele, and here it is forming a mu plus lambda chain rearrangement, and here if it again fails, it will form a non-productive allele again, and then after that it again will try to form a productive allele, otherwise it will die, or it will form a productive allele. So like this, it tries to minimize the wastage, and it tries to express only one receptor on its surface. And if it cannot do so, if it is seeing that it is producing multiple receptor on its surface, it will lead to the cell death. So uh, so this is all about B cell receptor rearrangement. Now we come to the T cell receptor rearrangement. So T cell, as I already have taught to you, like there are heterodimers. The T cell receptor is having heterodimers like alpha beta heterodimer or gamma delta heterodimer is present. So its structure is different from the B cell receptor. But it also contains this variable chain as well as the constant chain, similar to the B cell receptor. So this B chain was discovered simultaneously from two different laboratories. So from it is an interesting story how they found this B chain, and similarly after that in this procedure they found the alpha chain. So there were two scientists, Stephen Hedrick, Mark Davis, and their collaborators, who used a approach to isolate the B chain receptor genes from several mouse T cell hybridoma cell lines. So uh, what happened? Uh, so how they assumed 4 points, like while uh, discovering like what are the chains that is forming the T cells. So regarding the TCR genes, they assumed they had four assumptions, like TCR genes will be expressed only in T cells but not in B cells, and since these genes are encoded, these genes encode a membrane-bound receptor. The transcribed mRNA will be found associated with membrane-bound polyribosomes. So there are during the translation and like the transcription process, there will be a large number of ribosomes which will form a complex, and there, since PCR will not be secreted unlike BCRs which will form the antibodies, so the when after the transcription the mRNA will be found with the membrane bound polyribosomes. Then, like the immunoglobulin genes, the TCR genes will also have a variable as well as constant region, and the genes that encode the TCR will undergo rearrangement like that of the BCRs. Another group was TAC-MAC. Their group also used some other classical methods of molecular genetic analysis, but they also were successful in isolating the B chain gene of the human TCR. So like many in the world, everyone is working to, like many scientists are working. So it happens often like two or three groups work on the same problem. So whoever like can work fast and publish it will actually get more credit compared to the other people. So it is not like everyone is working in a unique problem. Everyone, like more than one group, can work on the same problem. Like here, two groups were there who discovered simultaneously about this B chain gene of the human TCR. Uh, so anyone had any question regarding any assignment, then they can tell. Then I will discuss that also. Uh, till week 8 if any problem is there in the assignment you can tell. We will study like how they actually did this experiment. So as I already told, they knew that the mRNA sequences which are encoding this TCR will be bound with the membrane-bound polyribosomal RNA. That is, the RNA sequences that encode membrane-bound protein from a. So what they did was they took a T cell hybridoma cell line. I have already told you about what is hybridoma last class. Not last class, maybe in one class I told. So hybridoma is like a culture of some hybrid cells that result from the fusion of any kind of cells with some cancer cells, so that what to say, so that they won't die, they will keep on proliferating. uh, they will be immortal cell lines. So from one T cell hybridoma means one T cell will be there which is hybridized with the myeloma cell line, and it has formed a T cell hybridoma cell. So what they did, they isolated the mRNA from the T cell hybridoma cell line. Similarly, they isolated some mRNA from B cells also. So after that, uh, they knew that 3% were in the membrane-bound polyribosome and 97% of the mRNA were in the cytoplasmic polyribosome. So from the 3%, what they did, they reverse transcribed into cDNA. Reverse transcription means from mRNA, you will get the DNA. Like normally we know that from mRNA, from DNA, we are getting the mRNA by transcription, then by translation, we are getting the protein from the mRNA. But here, you can reverse transcribe and you can form the cDNA. Where by using a reverse transcriptase enzyme, you can get the cDNA, which is the complementary DNA, which will not contain the introns of the genes, only contain the exons. So from there, from the mRNA, you can get the coding information of the DNA. So what they did was, uh, first they reverse transcribe this polyribosomal mRNA in presence of phosphate labeled nucleotides, so that whenever it is reverse transcribing, each of the phosphate of the cDNA will be labeled by the phosphate. So this cDNA which they made was labeled by 32P. Then they like mix this phosphate level T cell DNA with the mRNA they got from the B cell, and they hybridized it. So whatever regions are common with the cDNA and the mRNA of the B cell, they will hybridize, and they like they separated them, and they saw that there was some unique cDNA which were not hybridizing with the mRNA, and they chose only those mRNAs and cDNAs, and they removed all the other hybrids which were hybridizing with the cDNA. That means, like the which were hybridizing with the mRNA of the B cell means they were common. Those proteins or those membrane molecules were common to both T and B cells. So they discarded it. So they wanted to find the mRNA which is unique to the T cell. So what they did, they labeled the cDNAs. They used those cDNAs which were not hybridizing with the mRNA of the B cells, and they labeled these labeled cDNAs, as it was phosphate level, were used as probes to screen a library of cloned T cell cDNA. So they cloned many T cell cDNAs before, and they wanted to find that specific uh, receptor, and they took some T cell specific cDNA, and they hybridized it with this single stranded cDNA which were unique to the T cells. And this step confirmed that the T cell origin of the cDNA sequences and allowed the investigators to obtain cloned cDNA to use as reproducible probes. So, after that, they got two cDNA clones from a T cell library and that were identified as T cell and not B cell specific, and they used it to probe southern plots from liver cells, a B cell lymphoma, and from a series of T cell hybridomas. So here you can see they uh, took two type of cDNA, TM86 and TM90. So these are the two types of cDNA which they probed. Probing means like you are hybridizing the other DNA with the this cDNA and seeing whether it is unique uh, to that particular type of cell or not. So from the B cell lymphoma and liver cell, did not get any uh, different kinds of bands. But when they probed the cDNA with the T cell clones and because these liver cells and B cell lymphomas will not have will not have rearranged TCR genes. Only T cells will have the rearranged TCR genes. So what happened when they probed the cDNA with the different T cell clones from the T cell, they took some cDNAs and they probed it with this TM86 cDNA. They found that different bands were coming at different places in A, B, C, D, E, F, six different clones, suppose. So they were confirmed that these this why these different bands were coming, because this probe was binding to different regions of these clones. That means the regions has rearranged in different types of clones differently. So it is giving different size bands. But here you can see one cDNA they found TM90, which were giving the similar kind of bands as with that of the liver cells and B cell lymphoma. So they found that this is not a kind of this is not unique to the T cell, but this TM86 is unique to the T cell. Like this, they found that this is a B chain region segment which is unique to the TCR, and when you are having that probe or the CDN, it is binding to the different kinds of T cell clones and not the B cell lymphoma liver cells. Like this they found that B chain was is a part of the TCR. So here you can see the rearrangement, the germline organization of the TCL alpha, beta, gamma, and delta gene segments. So here there is C chain, constant region, as well as variable region. So each C chain segment is composed of a series of exons and introns. Organization of TCR gene segments in humans is similar. So here, this is a mouse TCR alpha chain and beta chain. In case of human also it is similar. So this is like the diagrammatic representation of the VD and J segments of the four families of TCR gene segments, showing the relative orientation with respect to one another on the three
You're
mouse
mute, mom.
chromosomes.
Cannot hear you.
Hello? Hello? Hello?
Yes, hello. The audio was off. The mic was off. That's why.
Sorry, I cannot. Okay, you could not hear me.
No, the audio was off. That's why I was asking you to unmute yourself.
Okay, okay, it was off. Okay, okay. You can hear, no?
Yes, I can hear you.
I wanted to ask whether any doubt was there uh, regarding any assignment.
No, no, not that really.
Okay. So this is how the T cell receptor segments are arranged. Like the VD and J segments are there, and it is similar in case of humans. These three are the mouse alpha chain and delta chain, and alpha, beta, and delta chain. And here you can see like this V regions is there, D regions is there, J regions, and constant region is there. Similarly, on all the cases, if it is a heterodimer between alpha and beta, it will form a alpha beta uh, receptor. If it is a heterodimer of other chains, it will form a heterodimer of with other chain. So the VD and this is the VD and J segments of the four families TCR gene to indicate that this many number of V chains are there. V alpha, you can see TCR alpha. Here, this many numbers of V alpha will be there, which can recombine with any J region, and it can form a that alpha chain. And here also, like BCR chains, it is arranged in clusters of V, D, and J segments. Here you can see this much, the like so many J's are also there, and so many V's are also there. Here B and delta receptor genes include V, D, and J segments. Alpha and beta receptor genes like Ig light chain genes only include V and J segments, of which there are again multiple different segments. So B and delta will contain VDJ segments, while alpha and gamma will only contain V and J segments. So all this will rearrange together to form different kinds of TCR or T cell receptor with different kinds of specificities. It is also similar, the process of this rearrangement is similar to the immunoglobulin gene recombination. Like PCR, I already told last class, it is uses some recombination signal sequences. Here also PCR gene use the same RSS or recombination signal sequences. But however, the arrangement allows DD rearrangement in TCR genes only in delta chain, that is enhancing the diversity of the CDR3 region. Here you can see like two D regions can join together and it will form the enhancing the diversity of the uh, TCRs. So here the TCR V and V segments are interspersed in a single chromosome region, and rearrangement of a functional alpha chain causes the deletion of the delta D, J and C segments, thus precluding the rearrangement of the delta chain. So here you can see, like in the alpha chain only, this delta region is also present, like the delta and the alpha chain are present on the same region. So whenever there is a need for the alpha chain, this delta region will be deleted, while whenever there is a need for the delta chain, the alpha region will be deleted. So the rearrangement of all these genes of the TCR will be similar. It will begin at the same time at the double negative stage of the thymocyte development. If successful gamma and delta V gene rearrangement occur, then T cell leaves the thymus as a delta gamma T cell. So, if gene rearrangement is unsuccessful but the cell generates a functional B chain gene, the cell undergoes several rounds of proliferation and then attains alpha gene rearrangement. If this is successful, then it passes through the thymic selection. It leaves the thymus as an alpha B T cell. So, like similar to the B cell, it will either form the alpha beta receptor or it will form the gamma delta receptor. So, after that, there will be a thymic selection process, as I already told regarding the B cell. Like the receptor should not react to the self antigens. The receptor only one cell will express only one type of receptor, and if all these criteria are met, then it will leave the it will pass through the thymic selection and it will leave the thymus and will go into the circulation. uh, However, in case of allelic exclusion, as I already told, in case of B cell, only one allele can produce one heavy chain and light chain, and another allele won't produce another heavy chain and light chain. Here, allelic exclusion is not complete in T cell as in B cells. In some cells, not all, obviously, some T cells can bear more than one alpha chain. So here, like B chain will express only one heavy chain and one light chain in a particular cell. Here you can have more than one alpha chain in a particular T cell. So allelic exclusion is not that much prevalent in T cells. So that's all for this TCR and PCR rearrangement. If anyone has any question, they can ask in the portal, or they can ask if any doubt is there regarding the assignment also in this rearrangement class in from both sir's side as well as mine side. And they can mail also, or they can ask in the portal also, or here also you can ask. So now we come to the cytokines. Cytokines, as you already know, that cytokines, sir has already taught, so I bet you guys know already that what are cytokines. So cytokines are a group of low molecular weight glycoproteins which are secreted by leukocytes, that is, lymphocytes, monocytes, macrophages, and other cells, and it mediate complex interaction involving lymphoid cells, inflammatory cells, hematopoietic stem cells, which is required for the development of an effective immune response. So they are very small, like small molecules, low molecular weight glycoproteins, and are secreted mainly by the macrophage, lymphocytes, monocytes, and they will have different kinds of uh, function in mediating the immune response. They are cytokines are also named as interleukins. There are many types of interleukins like interleukins 1 to interleukin 19, and they have other names like tumor necrosis factor and interferons. The chemokines are a group of cytokines that affects chemotaxis and other aspects of leukocyte behavior. So chemotaxis I have already taught. Like chemotaxis means it will attract other molecules to the site of infection. Like whenever there is a site of like whenever there is a entry of any pathogen and there is a breach in the epithelial layer, a group of cytokine can be released and it will uh, many cytokines will be released at that place, and there are chemokines which will attract different different like cells to that particular area, and that will clear the pathogens. So chemokines helps in chemotaxis. So in case of cytokines, there will be as like B cell, T cell receptors. There are also cytokine receptors which interact with these molecules, cytokine molecules. So the affinity of the receptor for the cytokines depends on the combination of the component chains. So there will be different types of, as you all know, that the receptors are protein molecules and they will be composed of different kinds of chains. Like as I already told, alpha, beta, gamma, delta. These are the four chains which will comprise the T cell receptor. Here also cytokines will comprise of different kinds of chain. Their receptor will compose of, and there are KD values, and there are different kinds of receptors like high affinity receptor, low affinity receptor, intermediate affinity receptor. So KD value means the dissociation constant, and it ranges from 10 to the power minus 10 to 10 to the power minus 12 molar, and hence cytokines can act at picomolar concentration. So you can understand the KD value means how the KD value low means there is a stronger affinity. So at a very small level also, this can interact with the receptor and can activate the signaling pathways. So binding of cytokines to receptors leads to increased expression of receptors and other cytokines that affect other target T cells, like the Th cytokines affect B cells, TCE cells, NK cells, granulocyte macrophages, and hematopoietic stem cells, constituting an entire network of interacting cells. So whenever there is a binding of the cytokines to the receptor, it can lead to the expression of more and more number of these receptors and other cytokines that can affect the other target cells like the Th cells, and these Th cytokines can affect other like cells. So there will be an interconnecting network that will activate one or the other cells. So cytokines can regulate the intensity and duration of an immune response by affecting activation, proliferation, differentiation, regulation of secretion of antibodies and other cytokines. So here you can see like the cytokines can like be like hormone. Cytokines also have this, like hormone will affect, can affect the same cell, or it can go and target the different cell far away from it. Like cytokines also has this ability, like when there is a inducing stimulus, this is a cytokine gene or the cytokine producing cell, it is producing cytokine. Here this there is a receptor. The receptor is binding with the cytokines, and there is a signal. There is a gene activation. There are different effects, as I told. There can be different effects like proliferation, differentiation, other cytokine production. So it will lead to the different kinds of biological effects. And here you can see there will be three types of action. One is autocrine action. This means the cytokine released from this cytokine producing cell will interact with the receptor on that particular cell itself and will induce a signaling in that cell. That is autocrine, means it is uh, activating its own cell. Then there is paracrine signaling. Like for example, the cytokines are produced in the nearby cells, like the in the extracellular tissue or space, and from there it is traveling to the nearby cell, and it is uh, inducing or affecting that nearby cell. Or there can be endocrine action, like it can be produced and it can move long distance and it can affect a distant cell. So as I already told, there are some similarities between the cytokines and the hormones. Both these are cytokines and hormones are induced by any kind of stimuli and then only secreted. Then these hormones are secreted by endocrine glands, and the cytokines are secreted by some special cells like Th cell, dendritic cell, macrophages. The cytokine can also affect the same cell or a nearby cell, while the hormone can affect a cell which is far away from its site. Then cytokine has a broad specificity for target cell. It can affect many types of cells, while hormone has only one or few types of targets. So the very low concentration of cytokine present in the interstitial space or blood is determined by sensitive assays like ELISA, that is sandwich ELISA, PAX, ELISpot assay using anti-cytokine monoclonal antibodies. So you can measure the amount of cytokines by different kinds of like assays like ELISA. ELISA is a technique by which we use antibodies to determine what kind of antigens are present in a particular sample. I don't know whether everyone knows ELISA, that is enzyme solvent linked immunoassay. So, enzyme linked immunoassay, and there are other techniques like FACS, which is fluorescent associated cell cytometry, and there is ELISpot assay, which uses anti-cytokine monoclonal antibodies. So, by using different antibodies which is specific to a particular cytokine, we can identify even very small amount of cytokine in a particular sample. The cytokines, as I already told, is very low in molecular weight, that is approx 30 kDa. They are differentiated into four families based on their structure: hematopoietin, interferon, chemokine, and tumor necrosis factor. So, what are the functions of the cytokines? One is they can regulate hematopoiesis, they can control cellular proliferation and differentiation. They can induce inflammatory response. They can help in the development of cellular as well as humoral immune response, and they can also heal the wounds. However, cytokines act in an antigen independent manner. They will affect whatever cells they encounter that bear the appropriate receptors and in a physiological state that allows them to respond. So it does not uh, like it will go and bind to any of its receptor, and it won't be like this much like a small amount of cytokine presence can actually induce any kind of cell which is having its receptor. So it is not that if the molecule is present in large amount only it will act in a particular cell. So does non-specificity of cytokines conflict with the established specificity of the immune system? So anyone can ask, so if a small amount of cytokine is produced and it can activate any kind of cells which is having its receptor, so it is like this is very much non-specific behavior. But does it affect the specificity of the immune system? The answer is no. Why? Because uh, it is like the cytokine receptors on the different kinds of cells are regulated. So not all the cells will be expressing the cytokine receptors. Most often what happens is only the cells which have interacted with an antigen will only express the cytokine receptor so that cytokines can act on those particular cells. And whether direct interaction between the cytokine producing cell and the target cell triggers cytokine secretion. Like thus, effective concentration of the cytokine are released only in the vicinity of the intended target. It won't be like too much, like too much long distance. Also, it cannot travel like as I told, like hormones, but it mostly like its function mainly occurs in the autocrine and the paracrine manner. So only in the vicinity, the cytokine which will produce can act on the vicinity of the target cells. So and also one most important thing is the half life of cytokine is very much low. So it ensures that they cannot travel far and far distances, and they will only be active till a limited time point. So these are the three different types by regulating the expression of cytokine receptor, by direct interaction between the target cell and the cytokine producing cell, and by maintaining the half life, the short half life. These cytokines will have a specific type of will act specifically, unlike the non-specific behavior of the cytokines. So by these three ways, the cytokine behavior is regulated. So, as I already told, there are different kinds of cytokine receptors. One is the immunoglobulin superfamily receptor, which is having the immunoglobulin domains. Here you can see these are the immunoglobulin domains. Example include IL-1, MCSF, c-KIT. Then these are the hematopoietin plus 1 cytokine receptors. Uh, they are the hematopoietin receptor family. This includes IL-2, IL-12, except IL-10. These are name of the different IL or interleukins. Then there is the class 2 cytokine receptor or the interferons. It is IFN alpha, IFN beta, IFN gamma, and IL-10. The different four types are there. Then there is TNF receptor family, TNF alpha, beta, CD40, nerve growth factor, PAS. Then there is chemokine receptors like IL-8, RANTES, MCAF, etc. and this is a G protein family receptor. So depending on the structure of the receptors, the cytokine receptor are divided into five families. These are having different kinds of domains, different kinds of structure. So there are different kinds of ligands or the cytokines which fall into different kinds of receptor families. So one of the more important um, receptor or one of the important cytokine is IL-2, and its receptor, that is IL-2R, we play a central role in the clonal proliferation of T cells. So the IL-2 receptor consists of three subunits: alpha, beta, and uh, gamma chains. The beta and gamma chains belong to the class 1 cytokine. Here, class 1 cytokine receptor family containing this motif CCCC, WXS, WS motif. And there are three types. This IL-2 receptor can be of three types, which is having three different affinity, three types of affinities for IL-2. So first one is low affinity monomeric IL-2 receptor. One is intermediate affinity and another is high affinity. So whenever there is a only one alpha chain present, here you can see it is having low affinity to that IL-2. But whenever there is beta and gamma chain present, here it is having intermediate affinity with IL-2. And when all the three are present together, alpha, beta, gamma, then it is having high affinity to IL-2. So you can understand by this receptor rearrangement and receptor expression also the cytokine behavior can be controlled. Here, as you can see, like the affinity constant is highest in case of this 10 to the power 11 molar in case of this alpha, beta, gamma, and it is then intermediate is having 10 to the power 7, then low affinity is having 10 to the power 8. But the dissociation constant is lowest in case of this, and then comes to the intermediate affinity receptor, then the low affinity receptor. Then, so as I already told, the lower the dissociation constant, the more the affinity. So depending on the types of chains, depending on the rearrangement of the receptors also, this cytokine level can be modulated. Like when there is a strong urgency of this IL-2 signaling, then all these three will be assembled together and there will be induction of signaling. Like much more strong induction of signaling compared to this low when there is only one alpha chain. So the gamma chain appears to be constitutively expressed on most lymphoid cells. Expression of the alpha and beta chain is enhanced after antigen has activated resting lymphocytes. So gamma chain will be constitutively expressed on the lymphoid cells. But after only that particular naive lymphocyte has seen its antigen, then only the expression of this alpha and beta chain is coming up to induce the signaling. The restriction expression ensures that only antigen activated CD4 and CD10 cells, CD8 T cells will express the high affinity IL-2 receptor and proliferate in response to physiological levels of IL-2. So, this restricted expression of the uh, receptors will only ensure that whenever there is a need only and whenever the T cells are activated by the antigen, the high affinity IL-2 receptor, that is all the three will come together and will proliferate in response to the physiologic levels of IL-2. This IL-2R alpha chain is expressed only by activated T cells, so it is often referred to as the TAC or the T cell activation antigen or CD25. A monoclonal antibody designated anti-TAC which binds to the 55 kDa chain is often used to identify IL-2R alpha on cells. So whenever this T cell is activated, this uh, IL-2R alpha is getting expressed on its surface, and it is often a marker for identifying an activated T cell. And you can also, its one other name is CD25. You can use an antibody against this CD25, that is an anti T cell activation or anti CD25, to isolate the cells, which is a to identify which are the T cells, which are the activated T cells. You can use this CD25 to see that this cell is activated, means this has already been exposed to the antigen. So using different kinds of antibodies, you can, as I already told, one like you can identify the concentration of the cytokines or the cytokine presence of the cytokine in any kind of sample by a technique which is the FACS or fluorescent associated cell cytometry. In that, by using that technique, you can use antibodies and can isolate cells which are having a specific marker on its surface. So by using, suppose, a particular antibody which is having binding affinity to a particular receptor on the surface of B cell, you can you can isolate B cells and you can isolate T cells by using another antibody. So from a population of mixture of many cells, you can use different specific antibodies which are specific to different kinds of like molecules on the surface of the cell to isolate different kinds of cells from a population of cells. So here, signal transduction by the IL-2 receptor because both the beta and the gamma chains, but only the trimeric receptor containing the alpha chain as well will bind with IL-2 with high affinity. So beta and gamma chains obviously is required for the signal transduction, but for the high affinity binding, alpha chain is required. So when all the three segments will assemble together, only then the receptor will be activated. Uh, so, general model of signal transduction mediated by most class 1, class 2 cytokine receptor. Here you can see just a signal transduction process where you can see this is a receptor, alpha and beta chain is there. Here cytokine is binding. Here you can see that when there was no cytokine present, it was separated. Whenever there is a binding of the cytokine here, then it is getting dimerized and which is activating the JAK or the genus kinases. These are the proteins which are like present near the receptors and it is getting these are the family of a tyrosine kinases and there is a phosphorylation on these genus kinases which is activating this genus kinases and which is then phosphorylating the receptor here. So this both this phosphorylation activates the other molecules. Like this also phosphorylase, this genus kinases will also phosphorylate the STAT molecules here in the SH domain, and here the STAT molecules will dimerize. It will go into the nucleus. It will transcribe the different kinds of genes. So these are the general uh, what is signaling which is done by the cytokines mostly. For example, STAT and JAK interaction with selected cytokine receptors during signal transduction. For example, IF and gamma cytokine receptor will activate JAK1, JAK2, and it will activate STAT1. And IF and alpha beta will also activate JAK1 and tyrosine kinase 2. will activate STAT2. Then IL-2 will activate the JAK1 and JAK3. It will activate STAT5. IL-3 will activate JAK2 and it will activate STAT5 here. IL-4 will activate JAK1, JAK3. It will activate STAT6. Then IL-6 will activate JAK1. Will activate STAT3. IL-10 will activate JAK1 and tyrosine kinase 2. It will activate against STAT3. Then IL-12 will activate JAK2 and tyrosine kinase 2. It will activate STAT4. So different kinds of cytokines will activate the different kinds of JAK and tyrosine kinases and JAK molecules, and it will lead to the phosphorylation of different kind of STAT molecules, which will then go into the nucleus, transcribe different kinds of genes, and induce the signaling. So there are different kinds of cytokine antagonists present, which will inhibit the function of the cytokines. For example, uh, antagonist means the proteins that will inhibit the biological activity of the cytokines, and agonist means which will enhance the activity of the cytokines. So, antagonists can act in two ways. Either they can bind directly to the receptor and they will directly to the cytokine, bind directly to the cytokine. So, if the antagonist is going and binding to the cytokine receptor, then the cytokine cannot go and bind to the receptor and it will not induce signaling. Or it can also bind directly to a cytokine so that it won't be able to go and bind to the cytokine receptor. So the best characterized inhibitor is the IL-1 receptor antagonist, which binds to the IL-1 receptor but has no activity. It cannot induce the signaling. It just will bind and block the place where the cytokine is supposed to bind. So when this IL-1 receptor antagonist is binding to the IL-1 receptor, it blocks both the binding of IL-1 alpha and IL-1 beta, and thus like it inhibits the signaling process induced by both of these cytokines. And these cytokine antagonists are also known to play role in regulating immune responses. Like this IL-1 receptor antagonist has been implicated in regulating the intensity of the inflammatory response. Sometimes the antagonists are good also because not too much cytokines can lead to too much inflammation in our body, which is not good. So someone should regulate these immune responses. So this antagonist can go and like regulate the different immune responses of these cytokines. IL-1 receptor antagonist has been cloned and is currently being investigated as a potential treatment for chronic inflammatory diseases. So, whenever there is a high inflammatory diseases, like for example, some autoimmune diseases or other kind of diseases or any kind of virus infection which is causing high kind of inflammation, or different normal arthritis or anything, there if you can actually give something which is controlling your inflammation, it will keep your inflammation in check. So IL-1 receptor is one of the potential molecule which is being currently investigated for a treatment. And there are many IL treatments also which are there in the market currently, which actually stops the inflammation in many kinds of uh, diseases. Like, for example, there are, I think, recently IL-2 is a treatment which is given for cancer. IL-2 treatment, or I don't remember the name, but some immunotherapy, IL-2 immunotherapy is given for cancer treatment. And people who are having cancer, it is affecting the whenever IL-2 treatment is given, it is stimulating our body's immune system to fight off the cancer. For example, I think melanoma treatment. But similarly, if you can use other treatment which is in in case of inflammatory diseases, if you are treating it with anti-IL molecules, then it can also decrease the inflammation. So cytokine inhibitors are found in the bloodstream and extracellular fluid. These soluble antagonists arise from enzymatic cleavage of the extracellular domain of the cytokine receptor. So these inhibitors and antagonists are found in the extracellular fluid or bloodstream. They will These antagonists will arise from the enzymatic cleavage of the extracellular domain of the cytokine receptors. The presence of soluble IL-2R has been used as a clinical marker of chronic T-cell activation and is observed in a number of diseases, including autoimmunity, transplant rejection, and AIDS. Like this IL-2R or soluble IL-2R is found in many kinds of diseases and used as a clinical marker for chronic T cell activation and is observed in a number of diseases like AIDS, autoimmunity, and transplant rejection. So, these cytokines are actually these molecules are activating our immune system and inducing different kinds of signaling. So there are many pathogens which have developed many strategies to bypass these cytokines also. For example, viruses, they have developed many strategies to inhibit this cytokines activity. Like they have produced, some viruses can produce cytokine homologs, which will go and bind to the cytokines or to the receptors. They also can produce cytokine binding proteins. They are also cytokine receptors. Then they can interfere with the signaling process. They can interfere with the cytokine secretion. They can also induce the cytokine inhibitors in the host cells. Like this, they have developed many kind of um, strategies to stop the cytokine activity. For example, Leporipox virus, which is a myxoma virus, produce a soluble IFN gamma receptor so that the cytokine will bind to this receptor and is unable to bind to the normal gamma IFN gamma receptor. Then there are several pox viruses which also can produce this IFN gamma receptor. Then vaccinia, which is a smallpox virus, produces the soluble IL-1 beta receptor. Then there is Epstein-Barr virus, which produces the IL-10 homolog, so that this IL-10 homolog will go and bind to the IL-10 receptor and there will be no signaling. Then this human herpes virus 8 will form a IL-6 homolog, also homologs of the chemokines MIP1 and MIP2. Then the cytomegalovirus will produce three different chemokine receptor homologs, one of which binds three different soluble chemokines like Lantis, MCP1, and MIP1 alpha. So you can see like how these viruses have developed, like how smart these viruses are. Like they are developing different kinds of like homologs or receptors so that they can bypass the activity of the cytokines. So uh, you can understand whatever I'm telling. If any problem is there, you can like stop in between and tell it. So, so T cells can be divided into two types. Th cells can be divided into Th1 and Th2 cells. That is T helper cell. We know there are two types of T cells: cytotoxic T cells and T helper cells. So, in T helper cells, you can divide into two different types of Th1 and Th2. For example, Th1 is responsible for many cell-mediated function. Example includes delayed type hypersensitivity and activation of the cytotoxic T cells. And for the production of opsonization, promoting IgG antibodies, like antibodies that bind to the high affinity Fc receptors of phagocytes and interact with the complement system, and for association with the promotion of excessive inflammation and tissue injury. So, this TH1 subset is having different kinds of functions, and this TH2 stimulates eosinophil activation and differentiation, provides help to B cells, promotes the production of large amount of IgM, IgE, and IgG isotypes, and support allergic reaction. However, the differences in the cytokines secreted by these two types of cells determine the different biological function of these two subsets. So, this TH1 and TH2 produce a different kinds of cytokine subsets which determines their function. So, you can see here the different kinds of cytokines which are produced by this TH1 and TH2 subset. Here, IL-2 is secreted only by the TH1, IFN beta, TNF beta, GM-CSF, IL-3, all these are produced by TH1, while GM-CSF to some extent is produced by TH2, IL-3 is produced, then IL-4, 5, 10 and 13 are produced by TH2 subtypes. Then, for elimination of a disease agent or its toxic products from a host, the immune response must induce an appropriate set of effector function. Example includes specific antibodies are required for neutralization of a soluble bacterial toxin, and also removal of the some intracellular pathogen requires mediated cell type cell type like cell mediated type cytotoxicity or delayed type hypersensitivity. So these are the different kinds of responses which is needed for different sets of pathogens. But how is this achieved? So there is a difference in cytokine secretion pattern among the T cells, Th cell subsets, which will determine what kind of immune response are made but in response to a particular antigen. For example, for total antibody production, if there is a need that the cell should produce antibody, then this Th2 cell is mainly responsible. For IgE production, this Th2 cell is responsible. For IgG2 alpha production, this Th1 is responsible. Then eosinophil and mast cell production, Th2 is responsible. For macrophage activation, delayed type hypersensitivity, TC cell activation, this Th1 is responsible. So, all different kinds of cytokines secreted by the Th1 and Th2 cells determines what kind of function that particular subset is going to do. So, cytokine mediated generation and cross regulation of Th subsets. Here you can see, for example, this there is a macrophage or dendritic cell. You know, macrophage or dendritic cell are APCs or antigen presenting cells. It will present the antigen which is entering into the cell in our body, and it will present this particular antigen to the T cell. And this T cell, after seeing the antigen or receiving the antigen from the macrophage or the dendritic cell or any kind of APCs, it will proliferate and differentiate to a particular T cell having a specific receptor, and then it will act on that pathogen. So here you can see via the MHC molecule, this macrophage or dendritic cell is presenting the antigen to the T cell or the T cell receptor. So this NAF CD4 T cell is receiving the antigen and it is getting activated. So it is producing IL-12, IL-2. So here you saw that IL-2 is produced by Th1 cells. So since it is producing IL-2, so this IL-2 will produce Th1 cell. This macrophage or dendritic cell also produces IL-12. So whenever this IL-12 also is produced, this IL-12 will lead to the generation of this Th1 cell, and this Th1 cell will then secrete different kinds of cytokines like IF gamma, IL-2, TNF beta, which will have different function like macrophage activation, delayed type hypersensitivity, certain opsonic or complement fixing IgGs. Then there is like this NAP CD4 T cell will also like this like if there is a excess amount of IL-4 in the environment, here you can see IL-4 is secreted, then this SNARE CD4 T cell will be Th2 subtype. If it is IL-2, it will go to the Th1 subtype. If it is IL-4, it will go to the Th2 subtype. And Th2 like in case of Th2 difference production, Th2 type of cell production, then this Th2 will also secrete other cytokines like IL-4, IL-5, IL-10, IL-13, which will lead to eosinophil activation and synthesis of IgE and some other antibody classes. So this IL-4 will give a positive feedback here, and this will synthesize more, like this will activate more and more of Th2 cells. Here also you can see this is giving a positive feedback here. And this is giving a negative feedback, and there will be no more uh, like this minus minus which this is a negative feedback which is produced, like no more cytokines needs to be produced, and not more amount of cells will go to the Th1 subtype and Th2 subtypes. So IL-4 can produce both a positive as well as a negative feedback. Here typically, the Th1 profile of cytokines is higher in response to intracellular pathogen, while in case of helminthic and allergic diseases, Th2 profile is more. Like when there is a invasion of any intracellular pathogen, then these cytokines by this Th1 site, like this Th1 cells are needed more, while in case of Th2 cells, only when there are any kind of allergic diseases or helmintic infection, this Th2 subtype will be more. Key cytokines produced by each subset positively regulate the subset that produces it and negatively regulates the other subset. Like here I told IL-4 is regulating the IL-2 subset, while this IL-2 or IL-12 is negatively regulating the Th2 subset. So there is a positive as well as negative feedback present, so that the amount of the subset produced is in a controlled manner and not in excessive manner, which can lead to more and more and inflammation. So there are different kinds of diseases, cytokine related diseases, which can occur. For example, there is if there is a defect in the expression of cytokines and cytokine receptors, then there can be a large number of diseases can occur. And for example, there can be bacterial septic shock. It is very common and potentially lethal. Develops a few hours after infection by certain gram-negative bacteria, including E. coli, Klebsiella pneumonia, Pseudomonas aeruginosa, Enterobacter aerogenes, and Neisseria meningitis. So, what happens in bacterial septic shock? There are bacterial cell wall endotoxin, which will stimulate the macrophage to produce IL-1 and TNF alpha, and not only produce, it will stimulate this, and endotoxin will stimulate macrophage to overproduce these molecules. So there will be septic shock, which will include the symptoms will include drop in blood pressure, fever, diarrhea and widespread blood clotting will be there in various kind of organs. Then the probable treatments are neutralization of the TNF alpha or IL-1 activity with monoclonal antibodies. Like you can provide the antibodies which will go and bind directly to this TNF alpha or IL-1, or you can produce or you can inject the antagonist. As I already told you previously, like this antagonist can help in decreasing inflammation. So this is really the septic shock is really a very bad disease, and it mostly causes death because it is, if not checked in the right time, the things go out of hand and people may die from septic shock. Then there is bacterial toxic shock, which are caused by superantigens. These superantigens will bind to class 2 MHC molecule, and the VB domain of the that is a variable beta chain domain of the beta domain, like the of the T cell receptor activating. Are there specific function than cytokines play in activation cells? Do they have a specification? Are there specific functions plays ones play in activation cells? Have a Yes, they can possess multiple function at the same time. And there are specific functions means, like I told, IL-2 can activate the Th1 subset. It cannot activate the Th2 subset. So some cytokines are there which can have specific functions. Some are there which can have more than one function, and some are having like only one function, and they have a specification on a particular cell means uh, they can mostly activate these T cells, then macrophages, dendritic cells only, all the immune cells only they can activate. So it can happen like one cytokine can uh, affect multiple cells, or uh, like it can have different kind of effect, like the effector function on different kinds of cells. Like one cytokine can play one function in one cell and another function in one cell. So uh, I will show you a slide. I had this slide, but now 1 minute. I hope you can see this slide. This I have taught in the previous class, so I didn't add it today. So, cytokine can have different properties, like they can have a property known as pleiotropic behavior, like it can have different effects on different cells. For example, in this IL-4 can activate, proliferate or differentiate B cell, can proliferate and differentiate TC cell, as well as it can proliferate mast cell. So it has different effect on different cells depending on the nature of the particular cell, and this is known as pleiotropic behavior. So another is redundancy, like two or more cytokines can play similar function. For example, here B cell B cell proliferation, these three cytokines can help: IL-2, IL-4, and IL-5. Then there can be synergy, like two cytokines, the function of two cytokines together will have a greater effect if like compared to the function of each cytokine. For example, here you can see B cell, IL-4 and IL-5. Here to combining together, their effects is more, and they induce the class switching to IgE, that is immunoglobulin E. It helps in the formation of the immunoglobulin E antibodies. Then, in some cases, the effects of one cytokine inhibit or antagonize the effect of the another. For example, this IL-4, this IFN gamma is blocking the activity of this IL-4 here and blocks class switches to IgE induced by IL-4. So these are the different properties of the cytokines: pleiotropic behavior, redundant behavior, synergic activity, antagonizing, and there is also cascade induction. For example, here this Th cells is producing this IFN gamma, which is then inducing the or activation of the macrophage, which is then again macrophage is producing IL-12, again which is in turn is activating the Th cell, which is again producing IFN gamma, TNF, IL-2, and other. So it is like uh, one cytokine on one target cell induces that cell to produce more and more number of cytokines. So these are the different properties of the cytokines. You understood what I told? Okay. So another type is the bacterial toxic shock, where these there are antigens or which are known as super antigens. Uh, so super antigens means they are antigens which are actually uh, like immunostimulatory molecules. They are those antigens are known as super antigens which can actually activate the immune system. They have a high like ability to induce the immune system, and they will majorly bind to the MHC molecule and to the TCR obviously, thus activating them. The antigens can activate a large number of T cell irrespective of their antigenic specificity. A number of bacterial superantigens are produced by Staphylococcus aureus, Streptococcus pyogenes, and Mycoplasma arthritis. There will be excess production of cytokines when there is a toxic shock like TNF alpha, IL-1, by since they are activating a large number of T cells, so there will be large number of cytokines which are produced. Then there is elevated concentration of cytokines that induce systemic reactions that include fever, blood clotting, and shock. So this is also not a good, like it is also a very bad disease kind of toxic shock. This both this septic shock and toxic shock, because this spreads very quickly and can lead to death if unnoticed. Cytokine activity is implicated in lymphoid and myeloid cancer. For example, there is abnormally high level of IL-6 by cardiac myxoma cells, that is a benign heart tumor, and myeloma and plasmacytoma cells, and cervical and bladder cancer cells. So here also in cancer also there is high level of IL-6 are secreted by this kind of cancer cells. Then there is one disease which is a Chagas disease, which is caused by Trypanosoma cruzi, and it causes severe immunosuppression. So uh, what you can do is Like you can co-culture the T cells with the trypanosoma cruzi, and then with labeled anti-TAC is CD25, which I told IL-2 receptor, and which binds to the alpha subunit. It showed that there is a 90% decrease in the level of the alpha subunit of the IL-2R. So that's why there is immune suppression, because it cannot activate the downstream signaling, and there is no activation of the TH cells, which lead to this immunosuppression. So these are the different kinds of diseases which actually are caused by the cytokines, and not excess. Anything is very bad. So whenever there is a like excessive cytokine, it is also not good. When there is a low cytokine, it is also not good. So the cytokine level should be maintained in the body in a level such that it can remove the pathogen and it won't harm our body. So any question is there? Okay. So this was all about cytokine. If any question is there regarding any assignments, you can tell me, or you can post in the portal also, or you can mail, and then I can clear your doubts next class. So next comes about the infectious diseases and vaccines. I will just briefly talk about this because next class only I will teach. Sir is now teaching about the different innate immune response against the different kinds of pathogens. So first, this is, you can see, just it is a woodcut showing sites for cowpox inoculation from an 1888 edition of some writings on pox diseases. Like, I hope you remember like how smallpox was cured by cowpox inoculation. So you can see here the top 10 causes of death worldwide among children under 5 are preterm birth complication, lower respiratory infection, asphyxia, birth trauma, diarrheal disease, congenital anomalies, then sepsis and infection, malaria, protein energy malnutrition, measles, meningitis, and these blue means they are communicable, they are maternal, perinatal, and nutritional condition, or these congenital anomalies are non-communicable disease. So you can understand there are so many diseases there, and so many things are there, like which needs to get cured, and so many people are suffering from so many diseases every day. So it is important to understand that different kinds of diseases or different kinds of immune response against these diseases, uh, uh, by the different kinds of pathogens. So first, we all know that the first line of defense is the innate immune response, and the first thing which a pathogen does is it breaches through our epithelial layers. So that is the first entry of the pathogen through our what is a body, and uh, there are barriers such as the skin and the mucus line surfaces, which acts as a buffer between the host and the infectious agents. So there are different kinds of barriers like epithelial barriers. There are these are the anatomical barriers which are present, which are the first line of like serving as a gate kind of between the host and the infectious agents. If it can open that gate, it can enter into our body. So there are sometimes what happens, these infectious agents get help from the other organism to circumvent this barrier. So in this case, a third party, which is called a vector, helps in to carry the infection from one person to another. So the vectors or the intermediate hosts can transmit a pathogen between one infected human and another, or from one infected animal to a human. So it need not be like one vector is there. For example, mosquito, most common vector which we can see around us, it can carry the pathogen. It can transmit the pathogen to our body, and from us also we can sometimes transmit that pathogen into another human. So mostly the infectious disease vectors are often the blood-sucking arthropods. Example includes ticks, flies, fleas or mosquitoes, which breach natural barriers like the skin with their bite and introduce the pathogen that can they carry directly into a susceptible host. So these vector-borne diseases account for approximately 1 out of every 6 instances of human infectious disease and are typically restricted to areas in which the intermediate host is found. Example includes malaria and Zika fever. So the first responder are the innate immune system, which are an additional barrier to infection, sometimes clearing pathogen without the need for an adaptive response. So first, this vector-borne infection, as you can see, in every 6 person, there is one person who is getting affected by this vector-borne infection. Sometimes it is not that much serious, but if it is serious, it can cause even death at its extreme level, because and that too, like for example in malaria or dengue, which is very much common in our tropical countries, uh, like these diseases are actually very much bad, and they are actually causing many deaths in like the season of the mosquitoes. When there is a high amount of mosquitoes, this also these diseases also increase. So we need to understand these diseases, and we need to understand how these diseases are circumvent by our immune system. So first responder, as I already told, is the innate immune system, and sometimes this innate immune system is enough to clear off the pathogens of the infection without even activating the adaptive response. So here you can see most infectious organisms enter through our mucosal surface, and like the respiratory, gastrointestinal, and urogenital tracts. However, they may breach skin barriers through a wound or by an insect bite. Once they have crossed the barrier, they exist in the sterile microenvironment of the body, where they can remain local or can spread through the blood and lymphatic circulation, making it a systemic infection. So examples are like there are 4 types of pathogen which can be found in the extracellular fluid, while intracellular parasites and bacteria may be engulfed by or enter host cells and remain vesicular. The viruses fuse with the plasma membrane enter the cytosol, from where they may insert their genetic material into the host chromosomes in the nucleus. Each of these locations allows for access to different detection molecules and may necessitate different effectors for elimination. So here you can see these are the different types of entry points through which the pathogens can enter. Like here, it can enter through the eyes, it can enter through our nose or mouth, it can also enter through our skin or through the urogenital tract. So it from different openings only you can see it can enter. After breaching the epithelial barriers, here you can see these are the bacteria, these are the parasites. There will be fungi, helminths, as well as viruses, and so here this viruses can be is extracellular, or it can enter into the cytosol or the intracellular, or this bacteria will be also entering intracellular bacteria. Then these helminths are there. Then these parasites will enter via the vesicles. So every pathogen will enter differently, and they will activate different kinds of responses. For example, in case of this mucosal, uh, these pathogens who are entering via the mucosal surfaces, For example, virus, bacteria, helminth, fungi, they will activate this signaling pathways. Then these extracellular ones like bacteria, helminths, and fungi, they will also activate these pathways. Then these bacteria and parasites which can enter through the vesicles, they can activate these Th1 cytokines, macrophage pathways. Then cytosolic means virus and some bacteria, they will activate this pathway. So different kinds of pathogen will enter through different routes and they will activate different kinds of pathways. So pathogenic disease mainly are four types of pathogens as I told: virus, bacteria, fungi, and protists. So virus include, you all know, HIV, which cause the AIDS, influenza, common cold viruses like rhinoviruses. Then bacteria include ear infection, pneumonia, food poisoning, which can cause this like Klebsiella will be there. Then Staphylococcus will be there. Many types of virus will be there. Then fungi, which will cause different this athlete's foot, ringworm, etc. Then this protease or the protozoa, which can cause malaria. These are the four types of pathogen which can cause different types of diseases. So I will talk about the different responses about this caused by this all these pathogens next week. So someone had a doubt. I don't know. She has not joined today. Uh, Anjali, she had one doubt. So I will clear it now. I will, if she joins next class, I will also tell like which of the following is not a function of a cytokine. So there are five options: cell proliferation, immune cells recruitment, cell death inhibition, oxygen transfer, and inflammation regulation. So according to her, why cell death inhibition is not a function of a cytokine, uh, is a function of a cytokine, because cytokines are known to induce apoptosis also. It can also even prevent cell death as well as it can also activate cell death. She told like she had a doubt like apoptosis, like it can cause cell death. Cytokines can cause cell death, but sometimes it has been seen that cytokines can also inhibit cell death, like inhibit apoptosis pathways. So it is not a function. It is a function of cytokine. Like it induces cell proliferation. It recruits immune cells. It inhibits cell death and it helps in inflammation. But oxygen transport is not a function of cytokine. So this oxygen transport is the correct answer for this question. So that's all for today. I hope in like if anyone has any question, they can ask or they can ask in the portal also. Okay, I hope you understood everything. So thank you.